All right, when we're looking at what you're targeting with bacterial medication specifically, you're going to target some specific structures and processes. You're going to target cell wall synthesis, protein synthesis, nucleic acid synthesis, metabolic pathways, and cell membranes. So here are some of the characteristics of these antimicrobials and some of the most common drugs that fall into each category. So targeting cell wall synthesis. Remember, when you look at the cell wall of a bacteria, it's unique because it has peptidoglycan. Plants have cellulose, fungi have chitin, and archaeans have pseudopeptidoglycan. So because bacteria have this and humans don't, it is a great target for medications because the majority of the time they actually have a high therapeutic index. So these are going to be drugs like penicillin, cephalosporin, and the beta-lactam antibiotics. Now, the limitation is they're really only effective against actively growing cells because you're interfering with peptidoglycan synthesis. They will weaken the cell walls, which leads to cell lysis. Remember, bacteria are in environments where the osmotic pressure is constantly changing, so they have that cell wall to help protect them from lysing. If you weaken the cell wall, you can burst. When you look at the drug bacitracin, it is toxic. That is why it's limited to topical applications. It actually interferes with the transport of peptidoglycan precursors across the membrane. Since it's toxic, it can only be used topically, but it is a very common ingredient in over-the-counter non-prescription medicine. Now, you can also target protein synthesis. You have to remember all cells make proteins. These medications do tend to be bacteriostatic, but you can create medications to target differences between bacterial and eukaryotic ribosomes. Remember, bacteria have a 70S ribosome, but eukaryotes have an 80S. What you have to be careful, though, is if you go after the ribosomes, mitochondria also have 70S ribosomes. So protein synthesis is not always a good target, and some of the medications that target ribosomes can actually be toxic and cause mitochondrial damage. When you look at the fluoroquinolones, they will actually inhibit topimerases. These help keep the DNA wrapped up correctly. So if they get damaged, basically you get these breaks in the DNA and it doesn't unwind correctly. You have rifamycins. These will actually interfere with RNA polymerase. And you have metronidazole. This only works in species that are anaerobic. You need the anaerobic metabolism to have a reaction with the drug because it converts it to an active form. Unfortunately, you can actually have mutations in the bacteria that allow resistance to these medications. You can target metabolic pathways. Targeting metabolic pathways is typically a pretty good choice because you target metabolic pathways humans don't have. So if you look at the sulfa drugs and trimethoprim, they actually inhibit different steps in the synthesis of folic acid. Well, here's the thing. We don't make folic acid. We have to get folic acid in our diet. Unfortunately, you can get resistance due to enzymes coded for by plasmids. So the sulfa drugs are the sulfonamides. They can inhibit a number of gram-positives and gram-negatives. They can basically bind in a form of competitive inhibition to the enzyme. And since humans do not have the enzyme, it does not cause an issue with us. Trimethoprim inhibits an enzyme at a later step. Now, humans do have a counterpart to that enzyme, but this medication doesn't really have any effect on it. And typically, you actually give a combination of both because they're synergistic to each other. And this is how we often treat UTIs. You can also target the cell membrane. Now, what this will do is actually cause the cell to leak, and it causes the cell to die. Unfortunately, we can only use these topically, and they tend to be not ideal because it also can cause our cells to leak, because they are capable of binding to our cells, even if it's to a lesser extent. So daptomycin will actually insert itself into the cytoplasmic membrane. This is used against gram-positives. You can not use it against gram-negatives because it's not capable of penetrating that outer membrane. You also have polymyxin B that you use with gram-negatives. Now, when you're dealing with Mycobacterium, not a lot of drugs are very effective. Remember, Mycobacterium is acid-fast. It has that mycolic acid in the cell wall, which prevents a lot of chemicals from getting in. It also grows very slowly, which prevents a lot of medications from working. There are 5 medications that are considered first-line antibiotics. They're given as a combination therapy, so you typically try to take 2 or 3 at a time to prevent mutants from evolving. They can try to target that mycolic acid. So isoniazid actually inhibits mycolic acid synthesis. That's why you're using it. The downside is isoniazid also is a known carcinogen. Now, if you're dealing with somebody with an infection that has resistant strains, they will take second-line antibiotics. The problem with these is they are less effective or they're more toxic. Now, we do have to test the susceptibility of bacteria to medication sometimes because you don't know which strain you have. So the susceptibility is unpredictable. So for this, you will calculate the minimum inhibitory concentration or the minimum bactericidal concentration. So the MIC is the lowest concentration that prevents growth in vitro. You'll actually use serial dilutions of the chemicals in growth media to look for turbidity. Inhibition doesn't necessarily mean that you have a successful treatment. You have to make sure that the level of drug can be achieved in a person's blood without poisoning them. And microbes with MICs between susceptible and resistant are termed intermediate. The MBC is the lowest concentration that will kill 99.9% of cells in vitro, determined from plate counts. These techniques tend to be very precise, but they're labor intensive and they're expensive. The disk diffusion method is a classical method used to determine susceptibility. So remember, you're going to grow bacteria on a plate and you're going to put filter papers with different types of medications. You will have what's called a zone of inhibition where there is no growth. That's only occurring if the antibiotic is killing the bacteria. Typically, you're looking for whatever drug creates the largest zone of inhibition. And unfortunately, different strains can actually react differently to the same medication. Now, we have commercial modifications of these tests. So we actually have growth rate turbidity cards that can actually do results similar to MICs in like a third of the time. We also have the E-test, which is a modified disk diffusion test. You actually have a strip that has gradients of antibiotics, and basically where the zone of inhibition intersects with the gradient, it lets you know what the MIC is. We can also measure the concentration of antimicrobial drugs in blood or other body fluids. So some of these are toxic. You have to actually measure how much medication is in the patient's system so that you're not poisoning them. So you can use diffusion bioassays where you are comparing known concentrations with the patient's samples. Basically, you have a standard curve, and you compare your results to the standard curve to figure out concentration levels. Let's pause here.